Okay, so I recorded that second video, the second makeup video that wasn't quite as bad, and I'm pretty sure I stopped on this slide, but I want to pick up here, even though this isn't like the world's greatest slide, because this is where we're going to be, what we're going to be talking about today. And I'll make sure you have a chance to ask me about concentration gradients. So when we go to lab today, we're going to actually watch diffusion happen in a beaker like I'm simulating here. So we're going to have, I should just do it in class. We're going to have an empty glass of water, and we're going to add some dye, and we're going to watch that diffusion happen. Okay, and just like if one of us set off a stink bomb in the front of the classroom, we're going to see those molecules spread from their contact point, like from their highest concentration, out into all of the rest of the fluid that has no dye in it towards their lower concentration. Okay. We're also going to look at some examples where we've taken some dye and sunk it to the bottom, and it's building up a concentration gradient. That's kind of what I'm mimicking here from the bottom. And so when you think about like taking a little pellet of dye and dropping it in and having it dissolve, as that dye dissolves, it's going to diffuse through the solution from the solid, which is like 100% dye, right? This is the highest possible concentration in this little dye nugget here. And it's going to diffuse out towards the place where there's like 0% concentration, right? Where the water is 100% clear. And it's going to move from that highest concentration towards that lowest concentration. And it's going to keep moving over time until we reach equilibrium, where the two are completely evenly distributed. Yeah.
I don't know if the thing is because I didn't see you like the small thing.
Oh, it's blinking down here. It said it was connected and it's blinking, so I think I'm okay. Yeah, and sometimes I do forget, mess up the sound, but it said it was connected, it said it's on, and it's blinking, so I think we're okay. So anyway, this forms a concentration gradient, and that concentration gradient just means uneven distribution. So we have high concentration down here, we have low concentration down here, and we're going to move from high to low, right? Most of the processes that happen in organism bodies happen in part because of a concentration gradient. That concentration gradient, the difference drives movement. Okay, we've got a lot here, not so much here. We want to be equal, and so things are going to move. Okay, the concentration gradient in this first picture we describe as being steep. We have a very steep concentration gradient, or a strong concentration gradient. And I know the picture isn't great, but we have a bigger difference between the most concentrated part and the least concentrated part than we do here, where there's less of a difference. Okay, and you'll see this more clearly in the bottles we have set up in lab today. You'll see like when there's a really stark difference, it's like black and white. That's a steep concentration gradient. If you have all the shades of gray between the two, then we've got a shallower concentration gradient or a weaker concentration gradient.
Is concentration gradient a force itself? Just a description?
I think it's more of a description than a force itself, but it can become a force. And so it can become a driving force, and that depends on how steep it is. The steeper it is, the stronger it is. I think sometimes it is a force. I want to say no, but we'll see some examples and that'll be clearer. It definitely can create electricity, it can create power, it can be a force. Okay. Usually that comes when we allow it to distribute freely. Okay, so generally speaking, simple diffusion, just moving like these dye molecules from high concentration to low concentration, is a really slow process. Okay, it takes a long time. If you think about those of you who drink tea or coffee in the morning, imagine like taking your sugar or your cream that you want to add to your drink and pouring it in and waiting for it to mix itself without sticking a spoon in it, right? 4 hours later, you're still sucking up sugar crystals from the bottom, right? It's really slow to have that happen on its own. But we can speed that up depending on what the characteristics are. Okay, so the rate that diffusion happens on, how quickly it occurs, depends on how big the molecules or ions are that need to be dissolved. Like a sugar crystal is pretty big, right? So it's got to dissolve first and then it can diffuse. And whether or not it fits easily, if there's any kind of a barrier, that size can be a big part of diffusion rate. The temperature of the solution makes a difference, right? You put sugar in iced tea versus in hot tea, right? Even if you stir it like crazy and add a lot of energy in iced tea, you might never get it to completely diffuse, right? And then the concentration gradient. The steeper the concentration gradient is, the faster diffusion occurs because there's a drive towards that lower gradient. If that gradient is pretty shallow or you have a weak concentration gradient, then diffusion is slower. So in this picture, we just have the same dye crystals dissolving in warm water versus cold water, and it's just the same amount of time elapsed, right? So warm water, the molecules are moving faster, right? So the diffusion rate happens faster because there's more movement inherent in that solution. Does this all make sense?
Actually makes a lot of sense.
Are you being snarky?
No, I'm just saying, like, for once, not you're putting slides there.
Oh, good. We'll call it a win.
Well, it's not like so.
Physical processes are easier. Yeah, okay. We're going to talk a lot today also about osmosis, right? And osmosis is just a special type of diffusion. It is the diffusion of water, okay? And typically when we talk about osmosis, we're talking about water diffusing across some kind of a membrane, okay? So we talked about our cell membrane as being selectively permeable, right? Meaning that some things go through easily and some things do not go through easily. And we looked at that. If you watched the lecture, I had a little example. I talked about small hydrophobic things like gases go through the cell membrane really easily. Small hydrophilic things like water can go through the membrane a little bit. But if you get to big ions or larger molecules, they really have a hard time going through that membrane, right? So when we're talking about osmosis, we're talking about movement of water against a selectively permeable membrane. And one of the things in our cells, water can get through, but it gets through at a really, really low rate because water's hydrophilic and the core of the membrane is hydrophobic. Right? So we have to have some sort of way to let the water through if we want it to go through fast. But the membranes are always a little bit leaky and some water is going to get through. So in these examples where we have like these beaker setups, let's just pretend this is some sort of synthetic membrane analogous to your cell membrane. It could be some dialysis tubing or something that's like making a barrier between the two sides. And so if we add, or if we have this situation here where we've added some dye molecules to each side, the purple dots, let's just pretend those are food coloring. If we've added some dye to each side, then the dye molecules are too big to go through the membrane. If they can't move, then the water's going to have to move. And the water can fit through this membrane at some rate. And so what happens here is that we have a low concentration of dye here and a high concentration of dye here. That means we also have a high concentration of water on this side and a low concentration of water on this side. So our water is going to move from high concentration to low concentration, just like our dye molecules would like to move the other way, but they can't, right? So our water is going to move towards the solutes, towards the dye, in order to reach equilibrium. And so in this case, in order for that to happen, we have to greatly reduce how much water is over here and get a lot more water on this side in order for them to have equal concentration. Does that make sense? Okay. So now we have the same amount of water per solute on both sides, but instead of moving the solute, we did that by moving the water. So I have to look it up again to be able to explain to you exactly what they do in reverse osmosis, but it's basically a filtering process that involves a very, 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 very fine pore filter that captures most of the things that are in water. And so there's lots of things that get filtered out of our water by our traditional like municipal water systems, but like there are a lot of things that just can't be extracted, like microplastics or like excess progesterone and estrogen from birth control pills, right? There's small things that can't get filtered out. Some of those can be captured by reverse osmosis, not all, but some. And so it's a much, much smaller filtration process. And I think that it involves setting up a gradient and forcing water through this filter in order to remove everything but the water. I'd have to look it up again to have it 100% clear. It's different than distilling. Okay. So when we talk about osmosis, osmosis is going to depend on the number of solute particles that are present as opposed to the kind of solutes. So when we're talking about these examples, we're looking at some pretty generic examples, but we're trying to get you to think about how this process happens in your cells, which are really complicated, right? So if you think about a cell, which I draw a little cell down here, and we think about, we've got our nucleus, and we think about all the stuff that's in the cytosol of the cell. We've got some positively charged stuff. We've got some negatively charged stuff. We've got a bunch of water. We've got some macromolecules. We've got some individual monomers. Right? There's lots of stuff in there. And if we're talking about having a gradient of concentration, that gradient is going to be relative to the total amount of stuff in the solution when we compare two solutions side by side. Okay? So when we think about movement of water towards some solute, it's going to be dependent on the concentration of that solute, not on what it is. Okay? So if we have, let me say this first and I'll give you an example because you're all like, what's she talking about? Like in the previous example, if we have a membrane across which water can pass, but these larger things, these ions and these molecules can't pass, then water is the thing that has to move in order to try to balance out that concentration gradient, right? And water is going to move towards the solutes because it's trying to make everything equally concentrated. It wants it to all be the same. And so if we have a high solute concentration, water is going to go towards that high solute concentration. When I was in elementary school, there was this horrible public service video that ended with dilution is the solution to the pollution, right? Like dump it all in the river, basically, because, you know, that's where we were at. Now we have microplastics in the ocean. So like that, let's not think that dilution is the solution to pollution, but it helps me to remember this, that like water is going to move towards the solutes and dilute them down, right? It's going to want to move towards those solutes and dilute them. And that's regardless of what kind of molecule is over here. So if we have a beaker and we have like 10% salt and 5% salt and 10% sugar, which way do you think the water is going to move back and forth between that barrier? Like which way is its net direction going to be? To this side? Like this? Yeah. Because this is 15% total and this is 10% total concentration. So it's going to follow the solutes to try to even it out. Okay? Whatever device works for you, whether it's a mnemonic about dilution being the solution to the pollution, or water moves towards the solutes, or everything goes from high concentration to low concentration, like figure out the system that works for you. But you're going to need to be able to puzzle this out. We need to be able to think about which direction should water flow? How are we going to build an equilibrium situation. And so water is going to go from high water concentration to low water concentration, which means it's also going to go from a lower solute concentration to a higher solute concentration and follow the solutes. You can think about it either way. Mm-hmm. Absolutely. So we're going to come back and talk about that in a minute. I thought it was the next slide. So even though I'm talking about there being a directional flow, water's always going back and forth, right? Anything that's able to fit through that membrane can go back and forth. But ultimately, the drive is to try to get towards equilibrium. The concentration gradient wants to get towards equilibrium. So there's going to be a net flow or a net gain to one side, even though some water molecules are still bouncing back through the other direction. Okay. So this process that we talk about when we start talking about this in organisms' bodies is osmoregulation. And so osmo, like osmosis, and regulation, like to regulate homeostasis, right? So this is basically water homeostasis. We talked about temperature homeostasis in your quiz and homework questions. We talked in chapter 1 about pH homeostasis or solute concentration, right? We have to maintain water balance. We have to maintain that balance on a cellular level and on a whole organism level. Okay, so osmoregulation is just the fact that organisms have to control their water balance, and they have to try to attain water homeostasis. They can't have their cells get too dry, and they can't have their cells have too much water in them. There's actually a limit. There's a too much also, where cells can start to burst because of too much water. So we've seen a lot of pictures of paramecium. I used them in our cell lecture where we were looking at microscopy in lab. This is a single-celled organism. It's a little ciliated organism all around the outside edge. It has a bunch of cilia that it uses to move, and it lives in a freshwater environment. We're going to talk about what that means in a minute, but basically this organism has more dissolved stuff inside its body than the fresh water outside of it, so water is constantly coming in. And so this little guy's living in a leaky canoe, basically. Water's always coming in, and he has to bail out all the time, or else his cell membrane might burst. So we talked about contractile vacuoles, and as we start talking about osmoregulation, I'm going to show you one in operation because it's kind of cool. And I'm pretty sure this does not have any sound. The sound is irrelevant. Okay, so I think you can see that there's two contractile vacuoles at least. There's one here and there's one here, right? Can you kind of vaguely make that out? So then watch them operate. So that one just emptied. The top one is filling, filling, filling. It's about to empty here any minute. The second one's empty the second time. Apparently the top one never actually empties. They tried to zoom in too much and they're out of focus. So yeah, it empties, and then as soon as it does that, you can kind of see that there's like a little bit of a star shape, and those are actually ducts that are collecting the water. I think I actually replaced this link with a better one. I think the better one is on YouTube. Yeah, the focus, the fact that the photographer is struggling to focus hurts my eyes. Yeah. Apparently that's what happens when osmoregulation fails. I can't seem to get out of this. Oh, hey, there's a lot of sound. Like, why can't I get out of full screen there? Yeah, plants actually have an ability to prevent themselves from exploding. We're going to talk about that in a minute. The overwatering problem is not explosion. It actually is more of a dilution problem and a loss of concentration gradient. Okay, so osmoregulation is maintaining water homeostasis. We're also going to talk about the tonicity of solutions. And so this could be any two solutions. We're typically going to be thinking about two solutions on opposite sides of a cell membrane. So the solution inside the cell and the solution in the surrounding interstitial fluid, or the solution inside the mitochondria relative to the solution in the cytoplasm, right? So solutions on either side of a membrane, okay? Tonicity basically just refers to the amount of solute in a solution. And so we're looking at sort of some measure of how much dissolved stuff is in that fluid. That makes sense. So when we think about cells and we think about organisms, when we think about what kind of solution those cells are bathed in, that might be a single-celled organism like our little paramecium who finds itself in a freshwater environment and is constantly taking on water, right? Or it could be a cell in a multicellular body that is bathed in the interstitial fluid around the cell. Okay, so I'm going to go through 3 slides. On the left, we have sort of a simulated beaker solution on either side with a membrane that's basically mimicking a cell, and then some solution outside of the cell, with the barrier being the membrane. And then we have what happens in an animal cell and what happens in a plant cell. So when we have solutions that have equal concentration on either side of a membrane, we have a situation that's referred to as isotonic. So the tonicity is the same, okay? That means they have the same amount of dissolved stuff on either side of the membrane, equal concentration of solutes on either side, okay? That means that we have the same number of dissolved particles, whatever those are, and we have the same amount of water, right? The same relative number of water molecules on either side of the membrane. In an animal body, like in humans, this is our happy place. Okay? Animal cells like to be bathed in a solution that has the same solute concentration as the cell. So here we have a red blood cell, and when the red blood cell is floating in an isotonic solution, some water is leaving the red blood cell, and some water is coming into the red blood cell, and overall, it has the same amount. Okay? It's neither gaining a mass or losing a mass of water. There's flow across the membrane, but there's no net exchange. So the cell stays at its normal shape and size. And this is how a healthy red blood cell should be shaped, this little concave pillow. Okay. In an aquatic plant, if we have something like our Elodea that we looked at in the cell microscopy lab that lives submerged in water, right? It actually doesn't want to be an isotonic solution. If it's in an isotonic solution, its central vacuole is not all the way full. So it's losing some water to the surrounding environment. It's gaining some water from the surrounding environment. There's equal balance, but it doesn't have any extra water to pump up its central vacuole. Because of that, the plant cell in this situation is what we call flaccid. It is not fully pressurized, and so the cell itself is sort of soft and doesn't have as much structure. So there's no pressure against the cell wall. And so instead of standing up really straight and having structure, the plant actually is going to be kind of floppy in the water. So this is sort of like when your house plants don't get enough water and they get droopy looking, right? That's because their central vacuoles aren't full. And so a plant cell in this condition is going to be okay, but it's not at its optimum. And so it's going to lose some of its structure. If we have a hypertonic solution, hyper means greater than average, greater than normal. The hypertonic solution has a higher solute concentration. So if we have our 2 sides of our generic membrane here, like this is inside the cell and this is outside the cell, if we have not a whole lot of dissolved stuff inside the cell and lots of dissolved stuff outside the cell, the water is going to leave the cell in bulk flow and go outside. Right? That has consequences for the inside of the cell. In an animal cell, as that water goes out to the outside, this red blood cell shrivels up. right? It loses all the fluid of the cytoplasm, so it concentrates down. The membrane kind of collapses on itself and starts to stick together. And it can result in cell death if it's not reversed. The cell is really unhappy. Drinking water helps us to maintain water balance, for sure. But it's a little more complicated than that.
Well, yeah.
But yeah, that's why we have to drink water, so that we have enough water in our body fluids. A plant cell in a hypertonic solution is also in really rough shape. So if we look in this picture, this is the cell wall of this plant cell in the center here. This is the plasma membrane. So what happens is the plasma membrane is surrounding the cytoplasm, right? So as that fluid goes out, the cytoplasmic volume shrinks, the plasma membrane shrinks. It actually completely detaches from the cell wall. So the cell wall is still there, just like when you go to buy a 2x4. It's made up of the remnant cell walls of the tree that you're buying. But the plasma membrane and the cellular contents shrink and separate from the cell wall. This is called plasmolysis. Plasmolysis becomes plasmolysed or shriveled. Okay. That's a very unhappy plant cell. Hypotonic solution has a lower solute concentration as opposed to a higher solute concentration. So now in this condition, inside the cell, we have lots and lots of dissolved stuff. Outside the cell, we have very little dissolved stuff, so water comes into the cell, right? In the animal cell, this causes the cell to bloat and bloat and bloat. And eventually, poof, the cell membrane breaks, okay? So what happens in an animal cell in a hypotonic solution is that we get lysis. Remember when we talked about hydrolysis? We said lysis to split or break. The membrane breaks. Paramecium exploding is a little bit of hyperbole because it's more like fizzling. Like it doesn't pop, it just kind of springs a leak. And so the fluid will start to burst through the plasma membrane. And so then the cell's contents are lost, cells not coming back from that. Okay. Cell death. Little tear, our dead red blood cell. Plant cells, an aquatic plant cell, super happy in a hypotonic solution. And that's because of the cell wall. The cell wall makes all the difference. So the water is coming in to the cytoplasmic fluid, and the cell is taking the extra fluid in its cytoplasm and it's pushing it into the central vacuole. Remember, that's its storage vat. So as it fills its central vacuole, the central vacuole grows, and then it pressurizes the inside of the cell. So the central vacuole is pushing on the cytoplasmic fluid, and it builds up pressure towards the cell wall. And at some point, when that interior pressure is high enough, it'll actually create such a rigid boundary of the plasma membrane against the cell wall that no more water comes in. So the plant cells, because they can interact with their cell wall, are actually able to prevent lysis. The cell wall is not going to break. So they pressurize against that cell wall and actually cut off the flow. So an animal cell will keep on taking water in until it breaks. The plant cell will fill its central vacuole, pressurize against its cell wall, and stop taking on fluid. So it can actually halt this flow of water. So a plant cell with a full central vacuole is said to be turgid. And in an aquatic plant, this would be the normal healthy position. This would allow the Elodea to sort of stand up in the water column and have rigid structure to its leaves. Does that make sense?
So if the other solution that was just a line, you could describe it as hypotonic.
Right. Yeah, we use these terms isotonic, hypotonic, and hypertonic when we're comparing solutions. If we had a single solution by itself, we would say it's 10% salt or it's 5% sugar. Like we would describe the solution with what's in it as opposed to trying to make a comparison. The hypertonic, hypotonic, isotonic terms are really comparative terms between two fluids. And in organisms, this is really important because we're constantly setting up these gradients of materials on one side of the membrane versus materials on the other side of the membrane. That happens within a cell and also between cells. Okay, so we'll spend the last bit of this class talking about transport processes. So I started off in the lecture that's on Moodle, the first one of many makeup lectures. The beginning of this conversation was saying that osmosis and diffusion were part of what we call passive transport. They don't involve the spending of energy, right? Facilitated diffusion is still a passive transport process, okay? But it's going to involve a helper to get through that hydrophobic core of the membrane. So facilitated diffusion involves using proteins that are called transport proteins to help molecules get through the membrane that otherwise can't pass that hydrophobic barrier. Okay? So things like polar molecules or charged ions can't go through that hydrophobic core. They get stopped. They get to about here, and then they get turned around. Right? So we use a protein to create a channel through that hydrophobic core so that these molecules can get through. Okay? Because this is a passive process, facilitated diffusion is driven entirely by concentration gradient. So solutes are going to move from a high solute concentration to a low solute concentration towards equilibrium every time, okay? Naturally. So I like to think of just all of these proteins as being transport proteins, but we actually sort of have two types of transport proteins. We have those that form a channel, and we'll just call those sort of generic transport proteins. And basically the protein itself, the way we've got this divided here, has a tunnel down the center. And that channel creates an opening from one side to the other side, very much like our gap or communicating junctions that we looked at. It's a way for materials to get from one fluid to the other fluid, regardless of their charge. So this area in the center here, this is all hydrophilic. Even though this out here is hydrophobic. So if this little molecule tries to go through here, it's going to be rejected and can't get through the membrane. But if it tries to go through here, it can go through the hydrophilic channel to the other side. Okay. So that's a transport protein. It creates a channel. These are sometimes produced in the membrane or taken away from the membrane. Some of them can be opened or closed and have gates on them, but it's a channel that allows things to move through. A carrier protein actually involves a shape change, and it actually binds to the solute that it's moving through. So it's a little bit more complicated, but it's still a passive transport process. It's still driven entirely by concentration gradient. So in this case, the carrier protein, instead of having a channel, has an opening that is the size of the solute. So this is sometimes very selective, like it can only pick the solute that's the exact right size and shape. And when that solute comes in, it actually causes the protein to change its shape. So in this case, this carrier protein is open here, sort of forming a V that way. When the solute comes in and binds, it closes at the top and opens at the bottom, and the solute moves through. Once the solute's moved through, it goes back to its original position, open at the top. And so it's going to move things from high concentration to low concentration by opening and pushing them through. Opening and pushing them through. Even though there's movement, it's driven entirely by concentration gradient. It doesn't take any energy. Okay. Does that make sense? So these are two ways to get things that are too large or too charged through the membrane. Okay. We also have a special type of transport protein that is known as an aquaporin. And aquaporins allow rapid diffusion of water. Okay. So a little bit of water can leak through. Osmosis always happens. But generally speaking, if we want to have actual bulk flow of water in like a mammalian tissue, we're going to have aquaporins. And so the aquaporins basically are special channels that allow one by one, like a single water molecule can go through the aquaporin at a time. So they can form like a little line of water molecules waiting to go through, but they can just move from one side to the other side. They only are big enough for water molecules, so larger things, bigger charged things can't get through an aquaporin. It's just for water molecules, but they can slip through. Some cells have lots of aquaporins, some cells have none. Right? So this picture up here at the top, this is actually a human egg cell that was genetically transformed to have aquaporins, and it's bursting. Like this is lysis right here. It's losing all of its cell contents out. And that's because it's not supposed to have aquaporins. And if you add them to it, it can't maintain water balance, and it just bursts. Okay. So many cells have aquaporins or don't have aquaporins as a mechanism for controlling water balance in those cells. Okay. All right. And lastly, we'll talk about active transport. Finish up chapter 4 finally. So active transport is going to require energy. And sometimes that energy can actually come from the concentration gradient itself. Other times it's going to actually involve, in most cases, expending ATP. But when we think about all these processes we've just been talking about, and moving solutes or moving water, thinking about concentration gradients, frequently we set up in our cells what's known as an electrochemical gradient. And an electrochemical gradient can be a charge imbalance across a membrane where we have lots of positive ions on one side and lots of negative ions on the other side, or lots of ions on one side and no ions on the other side. We end up with a charge imbalance. We can also create other types of concentration gradients where we have like a lot of solutes on one side and no solutes on the other side. We close all the gates and keep them apart. And then when you allow diffusion to happen, there's a lot of force and a lot of energy behind that. So if we move a bunch of ions to opposite sides of a membrane and we end up with a bunch of positive charges on one side and no charges on the other side, we actually have a little bit of voltage available. There's actually electricity there. And so that can power work. Right? So we can use that concentration gradient to power cell activities without expending ATP. They can be the energy source. And so sometimes when extra energy is available, we might use that available energy to pump materials to create these gradients and essentially store the energy in the gradient. And then when you let the gradient diffuse, that releases the energy. Okay, so these establishing and releasing concentration gradients is one way that the cell can store up and release energy. How quickly this happens and how easy it is to move ions depends on the distribution of charges and how strong the concentration gradient is. Okay, but we're going to see this as we move through the next few chapters. We're going to see this charge driving a lot of our processes. Okay, so active transport is transport that requires energy. We have to spend energy to do the transport activity. Okay. In this case, we actually move a solute against its concentration gradient or against an electrochemical gradient. So this is like building up those charges. Okay, we're pushing against the grain. It's like trying to move water up the river instead of letting it flow down. Right, we're going against the natural tendencies of the material of the solutions. Because of that, we have to spend energy to do it. Okay, so here we have another type of transport protein, which is an energy-driven transport protein. And this one is going to receive a specific solute. And so here, the shape of this protein is to receive 2 of these square solutes down here. The solutes are going to come in and bind, very much like in our carrier protein, except the binding itself doesn't cause a shape change. So the 2 solutes have come in and bound, and the protein is still facing this downwards, right? Now an ATP molecule that's carrying energy is going to come, and it's going to drop its phosphate group off on that, and in that transfer release energy to the protein. As it releases that energy, the protein changes its shape, and our solutes go to the other side. So here we have not very many solutes down here. We've got lots of solutes up here, and we spend energy so that we can add even more to that solution. The transport protein will return to its original shape when the phosphate group releases. Okay, so the phosphate group actually has to release, and then it goes back to its normal shape. And we use that release to cause the transformation. Signaling, sometimes just after it's shifted, it lets go. Depends. Anytime that we talk about a pump in animal tissues, this is what we're talking about. We have pumps like this in plant tissues too, but we're going to pump solutes across the barrier using energy.
So transport protein, passive, active.
Yeah, transport protein is kind of a generic term. Carrier protein or active transport protein or pump, carrier being passive, the active or the pump being active. So this is just sort of to compare the two. In passive transport, we're always moving from high concentration to low concentration. High concentration to low concentration, right? Whether we're going simply through the membrane in simple diffusion, whether we're using a simple channel transport protein or a shape change carrier protein, as long as we're going from high concentration to low concentration, it's passive transport. As soon as we go from low concentration to high concentration against the gradient, spending energy, then it's active transport. Okay. We have a couple of other processes that are active transport. Any process that involves moving large molecules is also active transport. So in order to take a macromolecule and bring it into the cell membrane or release it through the cell membrane, that's going to require energy. When immune system cells take in invading materials, that is going to require energy. So anything that's too large to cross the membrane, even with a transport protein, is going to have a separate energy-driven process. If it's coming into the cell, something is entering the cell, it's endocytosis, so it enters into the cell. So endo is to the inside. And so in endocytosis, the cell actually forms a vesicle around something and brings it inside. So here we have a little amoeba, and I'll outline the amoeba's body. Green might not have been the best choice. And what the amoeba is doing here is it's actually shaping its cytoplasm to create a little pocket, and food particles have accumulated in that pocket. And it's going to pinch around until it actually forms a vesicle, and it's going to engulf those food molecules into the inside of the cell. That's endocytosis. This is actually a white blood cell, and this is a bacterium. And so it's actually surrounding and engulfing the bacteria. And that is also endocytosis, moving something to the inside. Both of those are active processes. Thought I was going to be able to finish this chapter. I really did. Like 5 more minutes of this chapter. So after spring break, because next week is spring break, right? Woo, party down. We will take we'll take a few minutes to finish up chapter 4 after spring break. I've already posted the chapter 4 homework and quiz. They are due the week after spring break on Wednesday. So you have some time to get there. We'll finish this up on Monday, last few minutes, and then we'll have a double lecture on Monday. If you can stay, you can stay. If you can't, that's okay. But I'll do 2 lectures to make up for the one we missed this week. Between sick days and snow days, we're really falling behind.
That's the Monday.
The Monday after spring break. That.